Hello, and welcome to Thrilling Experiments. I'm Tony Thrill Hill, and I hope you're ready to do some thrilling experiments. Before we start, take a moment to breathe in and exhale. What did you just inhale? Oxygen, right? Where do you think that oxygen comes from? If you said from plants, you're exactly right. And that is what we'll be working with today. Plants. Way back in 1774, the English theologian and scientist Joseph Priestley discovered that plants have some pretty amazing properties. Priestley was studying the behavior of various gases, and he found that a candle placed under a jar would go out long before it ran out of wax. He also noticed that a mouse would soon expire if left under the glass. Priestley hypothesized that the candle and the mouse had somehow injured the air. Priestley wondered if a plant were placed in a jar with the mouse or the candle, could it restore the injured air? He conducted an extraordinary experiment to prove this hypothesis. To help us with our thrilling experiments, we need to bring in some expert scientific help. Please welcome June Cifuentes. Hi, June.
Hi, Tony. We are going to have some fun today. We're going to demonstrate how plants produce oxygen while they are busy making their own food.
You see, plants are very different from mammals and other animals. Animals are in a class called heterotrophs. They obtain their food from external sources, like plants or other animals.
That's right. And plants are autotrophs. They create their own food from water, minerals, carbon dioxide, and sunlight. A wonderful byproduct of that process is oxygen.
And we're about to see oxygen production firsthand. But one final thing before we get to the experiment. We need to remind everybody of the laboratory rules. Always have adult supervision when you're experimenting.
Plants as autotrophs.
So what do we need for the experiment, June?
We'll need a small candle, a small potted plant, 2 glass aquariums, and a stopwatch. Before doing this experiment, the plant should be in a sealed container, the aquarium, for at least 3 days and exposed to plenty of sunlight. This will allow it to produce oxygen.
What about the mouse? Don't we need a mouse?
Even though Priestley used a mouse, we'll get by using just the candle.
Okay.
We'll use the stopwatch to time how long the candle will stay lit inside the aquarium. Make sure you have a flat surface for the aquarium. A flat rubber mat works well. That way, when you cover the candle with the aquarium, no air will be able to escape or enter.
This looks airtight. I think we're ready.
Now light the candle and place the aquarium upside down over the lit candle. Start timing how long it takes the candle to extinguish.
Hey, it went out.
It took 12 minutes and 21 seconds. The flame consumed the oxygen in the aquarium. With no oxygen left to burn, the candle went out. Now relight the candle. Place it under the aquarium containing the potted plant. Record the time it takes the candle to extinguish.
Wow, it's still lit. That means that the plant is releasing oxygen.
Yes, this experiment proves that the oxygen supply in the aquarium is replenished directly from the plant. Pretty neat.
So, as we can see, without the oxygen provided by the plants, animals and many other organisms would not be able to survive.
Remember, when animals breathe or respirate, they take in oxygen and release carbon dioxide. When plants go through photosynthesis, they release oxygen.
That works out great for both plants and animals.
Exactly. It's called a complementary relationship. It helps plants and animals both survive.
Roots of plants.
Our next experiment involves growing seeds and the components of plant roots.
What do we need?
A plastic container about the size of a 9 by 13 baking pan, some paper towels, a spray bottle filled with water, and some radish or alfalfa seeds.
Okay, everything's here. We're ready to begin.
First, place several paper towels in the bottom of the plastic container. Next, spray enough water onto the paper towels to saturate them, but not too much. You do not want water to stand in the container. Now sprinkle the radish or alfalfa seeds over the wet paper towels. Place another layer of paper towels over the seeds and spray them with water. Remember, not too much. Next, place the seeds in a room with moderate room temperature and sunlight. You have created a perfect environment for the sprouting of your seeds. Within 2 days, we should have full-grown sprouts.
2 days?
You can't hurry plants, Tony.
All right. Here we go with that time-lapse thing again. I've got to admit, that was kind of fun. Anyway, let's check out the sprouts. Hey, they've weaved their roots right through the layers of paper towels.
Roots need a foundation, much like the foundation that holds up a house. As the seeds grew, they weaved through the paper for stability. The roots spread out to gather water and nutrients, so the plant can grow and develop.
What are the hair-like things on the roots?
Those are actually called root hairs, Tony. The plants use them to take up water from the soil. Pretty cool, huh?
Stem structure.
In the next experiment, we will see how most plants, except for algae and moss, take water and nutrients in and distribute them to the rest of the plant through an internal transport system. This system consists of 2 main types of veins, called xylem and phloem. The phloem carries glucose and nutrients, and the xylem carries water to the entire plant. We are going to focus on the xylem veins in this next experiment.
Okay, here's our volunteer. What's your name?
Hi, I'm Ashanta.
Okay, June, what do we need?
First, you will need several white carnations, some food coloring, water, several plastic cups, and a pair of scissors.
I got 'em.
To start, prepare several cups of water and mix each with a different food coloring. Set one carnation aside. Trim the other stems and place each in a cup of colored water. Split the stem of the last flower in half. And put each side in a different cup. It will be interesting to see what happens with this one. Now we have to...
Let me guess. Now we have to wait.
Yes, you're right. I'm afraid we have to wait several days for this experiment to work.
If you are trying these experiments yourself, you may want to do all the preparations at once. Then each day after that, you will have new results to study. But for us, it's time to zoom ahead another 3 days.
Wait, Tony, I've got a better idea. Let's just do this.
Hey, that worked great.
Oh my gosh, I can't believe what happened to these carnations. Check this out.
Isn't that amazing? The carnation that was in the blue water changed color. Look closely at the petals and you can see how the color was drawn into the plant with the water through the xylem veins. As you can see, the yellow didn't work well, but it isn't a very bright color. Let's check the split stem. Can you see where the 2 colors have been delivered?
Wow, I can see the different colors that were drawn up into the plant.
You can really see where each vein of xylem leads to within the plant.
Plant dissection.
For the next thrilling experiment, we will need some more flowers. Carnations or tulips work well. You'll also need a box cutter or utility knife, a diagram of a flower, tweezers, and a magnifying glass. A word of caution here. Please be very careful with the knife. Cut the plant, not your fingers.
I have everything for the dissection, Doctor.
Very funny, Tony. First, gently pull back the petals. The petals on a plant are like the landing lights on a runway. They attract bees to the flower. In a moment, we'll find out why. Once you've pulled back the petals, look inside the center of the flower. That thick part in the center is called the pistil. The long, tall structures next to the pistil are the stamen, which are the male reproductive structures of flowers. The stalk of the stamen is called the filament.
I have a question. What's that thing on the end of the stamen called?
The anther.
That's right. That's what I want to find out.
The anther.
Yes?
There you have it.
Have what? I need the answer.
If you mean the tip of the stamen,
Yeah, and I do.
it is the anther.
You know, I already forgot the question, okay?
Tony, the tip of the stamen is called the anther. A-N-T-H-E-R. It is the anther that produces the small reproductive cells known as pollen. This is the male portion of the plant that fertilizes other flowers.
All right, let me see here. Pistil. Check. Next to it, the stamen. Check. All right, the base of the stamen is called the filament. Check. And the anther up there at the tip makes the pollen.
Now take the box cutter and very carefully cut the flower in half, lengthwise, starting at the point where the stem meets the flower and go right up to the top of the pistil.
Uh-huh, so far so good. I think you've done a complete dissection.
At the base of the flower, you will notice some thicker green leaves. These are called sepals. Sepals protect the flower in its early stages of growth.
It looks like there are a lot of parts to the pistil.
Yes, there are. If you use the magnifying glass, you can see the inner organs inside the pistil. These are the female organs of the plant. The round base is the ovary, which contains small reproductive cells called ovules. They are like the egg cells in animals.
And the stamen next to the pistil is the male part of the flower that contains the pollen, just like you said before.
That's right, Tony. The top of the pistil is coated with a sticky substance that pollen grains will adhere to.
How do they move from the stamen to the pistil?
Remember the landing lights? Bees are attracted by the bright colors and the nectary, which contains the nectar that bees collect to produce their honey. As bees crawl down near the base of the stamen to the nectary, they transfer pollen to the pistil, and pollination occurs.
Nature finds a way. Well, there's so much to learn about plants, and we're out of time. But I hope everyone will continue to explore the world of plants. Thanks for being with us. Bye for now.
So long!
Bye-bye.
In the next experiment, we will see how most plants, except for algae and
Energy! Yay!
Thanks for the help. Who was I saying that to?
View all 10 titles in the Thrilling Experiment series.
Light,
Friction,
potential and kinetic energy,
Compounds,
human body,
Electricity,
magnetism,
Gravity,
plants,
Chemical reactions. All of the materials and equipment used for thrilling experiments were provided by Frey Scientific. Frey Scientific offers a wide selection of science equipment and materials to teachers across the country. Call Frey Scientific at 1-800-225-3739 or visit us on the World Wide Web at www.sciencescool.com.